My name is Tanner Dockendorf, and I have the pleasure of serving as Senior Manager within Aldevron's GMP Nucleic Acids business unit. Today, I'll be speaking about manufacturing considerations for successful gene therapy development, focusing on my observations and learnings of how beginning with the end in mind can help set up a program for success from the early stages. I want to start off by sharing a little bit about Aldevron. For anyone who's not aware of the organization or doesn't have a background knowledge, Aldevron is a CDMO focused on manufacturing nucleic acids in the form of plasmid DNA and mRNA, as well as recombinant proteins and monoclonal antibodies. Aldevron was founded in 1998 and has been focused, or was focused at that time, specifically on providing research-grade plasmid DNA manufacturing services. And over the years has expanded those services to include those mentioned previously, as well as expanded specifically the capabilities in the plasmid space to include clinical and commercial support in the form of our GMP-Source and GMP service levels. The organization is headquartered in Fargo, North Dakota, where all of our plasmid manufacturing operations still take place today. And we have capabilities at the facilities there to produce small milligram quantities through multiple tens of grams if required by our clients. Our current facility is a 70,000 square foot facility with 17,000 square feet of manufacturing space internally for GMP and then additional for GMP-Source. We are currently undergoing a pretty substantial campus expansion, however, to add another 189,000 square feet of facility, as you can see in the photo here. And these additional footprint should be online in the first part of 2021. Well, Aldevron has a variety of services that we offer. My talk today will center, as you're probably not surprised, around plasmid DNA manufacturing specifically. Over the past 22 years, we've worked with hundreds of clients and have expanded our services to support various uses for plasmid DNA in different end applications, ranging from critical raw materials to mRNA template material to direct inject API. Today, I'll hone in specifically on in vivo and ex vivo application of plasmid in the form of raw materials or intermediates for viral vector delivered gene therapy and gene modified cell therapy. These two applications do represent the bulk of our business from the manufacturing standpoint, specifically for GMP-Source and GMP. Everyone here today is likely, familiar with the rapid growth of the gene and gene-modified cell therapy segment of the industry over the past handful of years specifically. It's no secret these technologies are gaining traction and investment as a result of the successes and clinical commercial milestones we've observed by our colleagues in the space. If you think about Novartis, Spark, Kite, Bluebird, and others, this uptick has led to an increased trend or heightened focus specifically on the chemistry, manufacturing, and controls supporting these programs, among other things. This can be attributed to a variety of factors, and it's really hard to pinpoint, which is the most impactful. But I'll kind of go through all of these different components and start with expedited designation and accelerated approvals. Regulators around the globe have identified the need for a quicker pathway to move gene and cell therapies from concept to commercial, such as RMAT, RPDD, orphan drug designation, breakthrough or fast track designations, etc. These pathways really leverage more intentional and more frequent interactions between sponsors and the regulators in an effort to expedite the development workflow, and in some cases include things like rolling review. It's because of this that it's required for CMC strategies to be more established and well thought out earlier on in the life cycle of the program compared to what we might be used to with the rapid, or to keep track rather, with the rapid pace of this review and approval process. Coupled with this expanding regulatory presence, we also see an influx or uptick in the programs advancing to later phases of clinical development and even commercialization. a number of which are likely to continue, a number of the ones in clinical development, rather, are likely to continue to successful BLA filing. Scott Gottlieb and Peter Marks estimated last year that starting in 2020, we would be seeing roughly 200 new IND applications funneling through this review and approval process, and anticipated, or estimated rather, 10 to 20 approved therapies by 2025. We expect these milestones to continue to inform the CMC and development as a whole over the next handful of years. Not surprisingly, with this increased participation by the regulators and the influx of products funneling through these review processes, we've also seen a shift to more nuanced guidance and requirements coming from the regulators for both manufacturing and testing of cell and gene therapies. I direct you to the guidance issued in January of this year, 2020, by the US FDA, CMC information for human gene therapy investigational new drug applications. This has been a really valuable new resource for folks, and has given more concrete definitions around some of the requirements that we need to meet in this space. For a long time, it's been a bit of a gray area, so having this more structured, streamlined, and spelled out guidance has been quite beneficial. Shifting from regulatory items here, we're also seeing an influx of participants from outside of the gene and cell therapy space, joining organizations like small biotechs or even mid to large size companies, and bringing with them commercialization experience from a variety of disciplines. Think monoclonal antibodies, enzyme replacement, et cetera. This steady flow of fresh eyes and experienced minds and applicable experience leads to the continued review and evolution of CMC by leveraging these historical failures and successes that they have had in the past. Lastly, to combine two remaining points here, we have a growing understanding of the importance of a well thought out CMC strategy identified by continued mergers and acquisitions, license agreements, and other transactions, as well as an increased participation and substantial participation by investors even earlier on. Not only does the science need to be sound to secure these transactions and foster confidence by investors, but that CMC strategy also needs to be equally watertight and well thought out early on in order for these transactions to come to fruition. Now that I've set the stage a bit, I'll pivot and look at different avenues available for preparedness and specifics to consider within each of those. As a CMO, we've seen many different approaches when tackling these development strategies. Some have been really smooth and successful. Others have been bumpy and a bit of an uphill battle. And this slide highlights a variety of aspects of development to consider, which ultimately will better enable success. The most prevalent opportunities from our perspective to consider can be captured in three main categories. The first being construct design and elements of the plasmid DNA construct. Second would be the quality grade of material that you're using. And third would be the release specifications. I'll dive into each of these topics in the preceding slides. I want to make a disclaimer that this is specifically catered to plasmid manufacturing and that portion of the supply chain. And when you look at viral vector or cell product, there's a whole different set of considerations to think about and make decisions based upon that may be different than they are for plasmid. I'll start off with construct design and provide a high level of some of the key areas regarding this and how taking these into account early on can really enable future manufacturability and scalability for the long term. We'll start with taking a look at the GOI. So there's a variety of things to keep in mind when looking at the GOI. First of which is the importance of understanding the packaging capacity and ensuring you're not overstuffing your vector. And in conjunction with this, really working to reduce any non-essential sequences rather in the insert Streamlining this GOI can give you a benefit of a smaller GOI, which ultimately impacts overall plasmid size and thus yield and number of copies per E. coli cell. Another really critical thing to think about when assessing a GOI is retention and stability of ITRs, or inverted terminal repeats. Specifically in the case of AAV-delivered gene therapy, it's really quite critical and must be demonstrated by restriction digest analysis, very commonly SMA1 or AHD1. Sequencing is the gold standard for ensuring retention of these plasmid components. It can be difficult, but is absolutely doable. But confirming this ITR stability early on is highly encouraged. Otherwise, it will be an uphill battle for the lifetime of the project if it's not improved. Lastly, one thing to consider is the impact GOI expression may have on the growth of the E. coli expression system you're utilizing. Certain GOIs can prove to be toxic to the cell that they're being expressed in, which leads to suboptimal performance from a yield and productivity standpoint, which can then subsequently impact impurities downstream and really impact the overall purification process that you might be looking at. Next, we'll take a look at the origin of replication, which is another key driver of specific yield, or the number of milligrams of plasmid you're getting per gram of E. coli. Like I mentioned previously, this specific yield has a direct impact on the level of impurities seen downstream, things like endotoxins, as well as residual host components like genomic DNA, RNA, and protein. In the simplest terms, a higher specific yield gives you a lower ratio of impurities to plasmid. The alternate is also true, where a lower specific yield will give you a higher ratio of impurities to plasmid. So main takeaway here is that a more effective origin of replication will enable scalability of the plasmid manufacturing process. And it becomes really critical when looking at expanding to larger patient populations as you progress through the clinical development pathway, as well as when you're looking at indications that require larger genetic payload, larger doses of virus, whatever that may be. Lastly, we'll take a look at antibiotic resistance. There's been a really significant shift in the design of constructs, or even the retroactive modification of constructs, to include kanamycin resistance rather than ampicillin. We do still see some lingering usage of ampicillin for a variety of reasons. However, with the keen eye the regulators have on residual beta-lactams, it's continuing to drive the early introduction of kanamycin into the process. And then for an even more risk-averse approach, there are folks that are looking to remove altogether antibiotic resistance within the plasmid, and thus the antibiotics introduced in the manufacturing process via technologies like sucrose selection that companies like Nature Technology are introducing. When looking at these type of technologies, it's important to balance this benefit with the potential hurdles that may introduce as well. Things like impact to yield or cost, general licensing of a technology, and the subsequent freedom to operate. We'll pivot into some quality considerations as well. Quality is one of the most frequent unknowns we hear about from our clients. There are a number of considerations to make when determining what quality of material is appropriate for your downstream application, for the product, for the phase of study, etc. And there are equally as many, if not more, options out there in the industry from a manufacturing standpoint that you can leverage different intermediate quality levels, different flavors of manufacturing, etc. But it's really important to think about the different components that can lead to an informed decision of quality. The first being patient impact. Truly, the impact of the patient is one of the key factors when assessing quality of material. Look at proximity, i.e., how many steps removed is the plasmid going to be from the patient. This will impact the risk profile of the material and drive the decision. That, coupled with the route of administration and the dose size, can also impact your decision. Typically, we are seeing folks, the closer the plasmid is to the patient, really looking at a higher quality grade or that GMP level. Aside from patient impact, your clinical plan is really critical as well. The clinical development plan can impact and inform quality requirements, especially when looking at where you are currently, where you plan to be, and when you plan to be there. Circling back to some of those accelerated approvals or expedited review designations, this can really shift the needle as to where and when you need these higher quality materials and when you need to introduce those into the process. And to that end, we find that many people are looking to build in efficiencies as early on as possible, whether this be driven by those accelerated approvals or expedited review designations, or generally just to mitigate timeline or quality risk. We see more commonly the introduction of full GMP components earlier on in the process. So think things like introducing GMP master cell banking right away with the early phase work, then having that GMP cell bank to go back to when you're ready to move into later phase or GMP manufacturing. Or on the other end of the spectrum, we have folks who will leverage GMP from the jump and utilize GMP material for early phase through commercial. Similarly, scaling up early on can also offer efficiencies downstream, wherein your manufacturing process is already set at a scale which can support your expanded larger trials or even be at the same scale you'll need to support commercial. With this approach, you can potentially leverage a single larger batch early on to meet the needs of early phase as well as potentially pivotal trials, and then have that process locked in and ready to go for commercial when the time comes. I will pause and make an umbrella statement here about quality that it's really critical to have an open line of communication with your regulatory team if you have one, or with a regulatory consultant if you're taking that route, and really just interact with the regulators and the agency early on to receive guidance on what is appropriate for your product and for the phase of study that you're at. We'll show here also an Aldevron specific kind of high level overview of quality and phase of development and what we see most frequently with our clients. Like I mentioned, there's a variety of intermediate service levels available out there, and ours is termed GMP-Source. And what we most frequently see is clients leveraging that GMP-Source manufacturing service level for that early phase requirement, but potentially layering in some GMP components earlier on, like I mentioned in cell banking, or bringing all of that GMP fully upfront and utilizing that for all of the project components from that early phase. But ultimately, all of our clients are shifting to that full GMP manufacturing as they progress through late phase and really seriously gearing up for commercial. I'll move next to some considerations around release specifications and quality control. The heightened focus on CMC also extends into release testing and specifications. Per the most recent FDA guidance on CMC, it's recommended plasmids be tested to ensure identity by sequence verification as well as restriction digestion. I'll refer back again to the comments I made previously about ITR retention and confirming those via restriction digest with SMA1 or AHD1, as this is a really critical point. Aside from these usual suspects of identity assays, the agency has also nodded to the importance of understanding the ability of your process to remove residual impurities, thinking about things like endotoxin or residual host genomic DNA, RNA, or proteins. This will help in establishing comparability for future production runs or comparability if you're making any modifications to the process later on. We also see a variety of requirements in the realm of safety assays, most notable when assessing sterility testing. Determining which method or which approach to employ is really important, as it does impact manufacturing strategy as well as overall program timeline, as some of these assays are quite time intensive. There's a few options to look at in this space. Of course, the traditional USP71, as well as rapid sterility, and then also bioburden. It's up to you, the sponsor, to determine which is going to be the appropriate choice. But we have far and away seen that sterility is the most popular approach. And recently a shift to rapid sterility from the traditional USP71, again, getting to that point of more efficient timelines. To sum it up here, it's really important to plan for the long term when setting initial specifications, as the process for changing these midstream is often challenging from both a cost and a time standpoint if you don't do it with appropriate justification. However, having said that, there's also an appreciation and understanding that setting these specifications initially for early phase studies should allow for some refinement further on as you do receive data from additional manufacturing runs or other variables that may impact your release criteria. So it's really a balancing act here of setting specifications that allow for enough flex later on, but also not being too stringent on the front end and, painting yourself into a corner with some of these specifications that are potentially unrealistic to carry on through the whole development pathway. Lastly, I want to give a nod to a saying we have here at Aldevron: fortune favors the prepared. It's not uncommon for groups to overlook plasmid supply as they rush to secure capacity for downstream manufacturing, whether that's viral vector manufacturing or cell therapy manufacturing or some other modality. But it's often that groups get in a real bind if they overlook the plasmid and then try and shoehorn plasmid manufacturing into their vector production timeline or whatever the downstream is. So I hope that this presentation has given kind of some insight into the complexities and components that go into setting up for success with plasmid manufacturing, and I've also outlined here some high-level buckets of different components feeding into the plasmid manufacturing process, and really things to consider, everything from even scoping out the project at the front end to putting in place agreements like an MSA or quality agreement, and then looking at different components you can layer in, optimization and engineering, actual stability studies on the back end that really impact project scope and timelines. There's just a lot to think of, and so we'll leave you with the sentiment of remember the plasmids, please. So this rounds out my talk. I hope that at a minimum, this has given some fodder for thinking about different considerations to make when preparing for manufacturing. And I'm certainly happy to take any questions, and feel free to reach out to me. And with that, thank you very much.